Good morning, cohort students. Today we are learning about DNA. We are continuing our DNA transcription, translation, protein synthesis, genetics unit. And today we're going to be talking about DNA basics. Some of this will be a review because y'all did great on the DNA labeling assignment that you did last week. But this information will help you on today's DNA basics assignment that is due Tuesday by 11:59 p.m., as well as your check for understanding, which is coming later in class. So we are going to be talking about the discovery of DNA as well as the structure and the function of DNA. So the first thing that I want to talk about is the structure, okay? DNA is a double helix, okay? And you guys created a 3D model of this double helix in engineering. So I think it would be cool for you guys to hear a little story about the background information on this double helix. So Rosalind Franklin is a woman who took the first picture of DNA. She was the first person, the first woman to look at DNA and look at its structure, find its components, and she took a picture of it with a very strong microscope. This photo is called Photo 51, and it's right here. Okay, you can kind of research what these mean here, but you can kind of see this here as well as this, like the cross of it, is that like rotating structure. Okay, so if you were to take DNA, the structure of that 3D molecule, and look at it from the top down, okay, that's kind of what she's seeing. So she was working in a lab, and her lab partner, Wilkins, he was also doing his own research on DNA. And he saw her photo. Of course, they're working in the same lab. They are exchanging, thoughts about DNA. And he took her picture and he shared it with Watson and Crick, who are the names that you normally hear when you are thinking about DNA, structure of DNA, because they wrote a paper in 1953 about the structure of DNA and included this photo in it. And they ended up winning the Nobel Prize in the 1960s for the structure of DNA and kind of finding its components. And so Rosalind Franklin didn't quite get the acknowledgement that she did deserve with that, since she took the first photo. And if this is an interesting story to you, or you're interested in women in science, there's a National Geographic article that's linked in these slides, and you have access to them, and you can read about her as well as some other figures in science who didn't quite get the acknowledgement that they deserved. So I find that very interesting. But DNA is a double helix, okay? And you guys did create a molecule of that. I saw some of them and they are wonderful. So deoxyribonucleic acid, that is what DNA stands for. Now, DNA has a major component to it, okay, which includes the smaller components that make the entire molecule. So the building block, or the monomer of DNA, is what we call a nucleotide, okay? Now, a nucleotide has 3 components to it, and here's a picture of it right here. And many of these nucleotides, so if you took this structure and you stacked it on top of each other over and over again, it would create the whole molecule. And we'll look at a picture a little later of that. But a nucleotide has a 5-carbon sugar, okay, that is the structure right here. And there is a carbon on each kind of edge of this, or each corner of this pentagon here. Okay, you don't really need to know that until chemistry, but this is a 5-carbon sugar, okay? There's also a phosphate group attached to this sugar. These 2 things make up the backbone of DNA. And then we have our last component of our nucleotide is what we call nitrogenous base, okay? Now, there are 4 types of nitrogenous bases. You should have this in your notes already, but it is adenine, cytosine, guanine, and thymine. Okay, so we're going to talk about those next. But these 3 components together are a nucleotide. So here's a picture of DNA, and you can see its components a little bit better, right? So here's your double helix 3D structure, Okay, and you can see that kind of down here as well. So this blue portion, this blue ribbon portion, is what we call the backbone of DNA. Okay, so you can see it kind of unwound here. So when you think of a DNA molecule, it is kind of a spiral twisted molecule. Okay, in order for us to think about its components, we're going to think about it unwound. Okay, in this kind of ladder structure here, where you have your alternating sugars and phosphates as your backbone, and then your nitrogenous bases in the middle. Okay. So let's look at these components. You have your nucleotide here. Okay, so this box that is around the nitrogenous base, and this one happens to be guanine, your sugar here, your 5-carbon sugar, and then your phosphate. Okay, so those three things together, whenever you see something circling all of them, that is your nucleotide. Okay, so notice each nucleotide stacked on top of each other makes an entire DNA molecule. You also have your base pairs, and this is labeled as these matching letters in the middle. I'm going to go over in just a minute why these are matched together or paired together, but you will always have adenine with thymine, and you'll always have guanine with cytosine. Okay, so colors and the way that these nitrogenous bases are paired or matched, it does matter. In between your nitrogenous bases, they are bonded together by what we call a hydrogen bond, okay? Now, we're not going to get into the chemistry of that, but there are different types of bonds in chemistry. There's covalent bonds and ionic bonds and hydrogen bonds, but what you really just need to know here is that it is the hydrogen that is bonded together to create this bond that happens right here at the base pair, okay? Next, I want to talk about the two types of bases. So we have 4 nitrogenous bases, okay, but they are categorized into 2 kinds of bases. You will have what we call purines and pyrimidines, okay? Now, I'll tell you guys the way that I remember which ones are which in just a moment, but what you really need to know is how big the structure is. So purines are double ringed bases. Okay, now what I mean by that is molecules, when it comes to molecules in your body, when we get down to the chemistry, are made of rings. Okay, so you saw this 5-carbon sugar. Okay, your purines are going to have a double ring base. So here's 1 and here's 2. Okay, so you see a pentagon and a hexagon here. Okay, that's its chemical shape, pentagon and a hexagon for guanine and for adenine. If there are 2 rings, they are known as purines. Okay, the purines are adenine and guanine specifically. Okay, your pyrimidines are single ring bases. Okay, so whenever you see a single ring, this is just a hexagon here, that will be a pyrimidine. And your pyrimidines are going to be thymine and cytosine. Okay, so it will be important that you remember those. Purines, adenine, guanine, pyrimidines, thymine, and cytosine. Now, the way that I remember this is that it pairs with the opposite. Okay, so a purine is a shorter word, but it is a double ring. Okay, so if I'm looking for a purine, I automatically think 2 rings because it's kind of the opposite. And a pyrimidine is a longer word, but it only has 1 ring. Okay, that might help you, maybe not, but a pyrimidine will only have 1 ring, purines 2 rings. Now, there's a rule when we are pairing our bases together. Okay, our nitrogenous bases, which connect at the middle with those hydrogen bonds, they will pair together. This rule is called Chargaff's rule, and it states that adenine and thymine always pair together, and guanine and cytosine always pair together. Okay, this means, if you notice, you will always have a purine paired with a pyrimidine. Okay, so a purine and pyrimidine will connect at the middle with that hydrogen bond. Adenine always with thymine or A and T. Guanine always with cytosine or G and C. Okay. What is important to know about this is because of this pairing, there is always the same amount of each in the DNA molecule. For example, if you have 30% guanine in a DNA molecule, that means that you also have 30% cytosine, okay, because cytosine is paired with guanine, okay? Now, if you know the percentage of one nitrogenous base in the molecule, you can then find the percentages of all of the nitrogenous bases in the DNA, right? So I have 30% guanine and 30% cytosine. That equals 60%. Okay, that must mean that I have another 40% that is then divided between my adenine and my thymine, meaning that I have 20% each there. Okay. So I wanted to show you the difference between RNA versus DNA. Okay, we're going to be learning about protein synthesis, which is taking your DNA and translating that information into a protein to be used in your body, which is constantly happening inside of your cells. So you'll need to know about RNA as well. DNA is deoxyribonucleic acid, whereas RNA is ribonucleic acid. Okay. RNA is only one strand, okay? So you'll see here we have our double helix as my DNA here, and just a single strand of RNA, okay? So RNA is going to be just one strand. They both have base pairs in the middle, okay? But RNA will only have them again on one side. And in RNA, we have a special nitrogenous base that is in RNA that is not in DNA. And you'll have to know this for your assignment today, okay? So when I am matching my letters or my bases together in my RNA, instead of thymine, okay, which is available in my DNA molecule, RNA will have what we call uracil, okay? Uracil, notice, is only one ring, okay, so that is a pyrimidine. And in my RNA, my G's and my C's will still match together, okay? But instead of A matching with thymine like it does in DNA, A is going to match with uracil. Okay, so whenever I am matching my base pairs and I see an A in my DNA, it means that it will pair with a U in uracil. And this is true for all RNA, okay, because there are different types of RNA that we'll learn about in protein synthesis. So remember that thymine is only in DNA and we have uracil instead in my single-stranded RNA. All right, lastly, I wanted to take a look at this structure because you are going to see multiple different pictures, different diagrams, and things like that when it comes to DNA over this next week. And then also when we return from break. So you need to be able to look at different structures of DNA and kind of see what that is in your mind, okay? So for example, this is kind of what you saw in your DNA labeling assignment, right? You saw the actual chemical structure of things. You saw those 5 carbon sugars here, okay, and those phosphate, those rounded phosphate groups and the hydrogen bonds in between, okay? So you'll notice that you might see like a chemical structure like this, but you also might need to label something like an actual double helix, okay? So it will look differently. So here's my chemical kind of structure, and you can see your A's, T's, G's, and C's here, and they are labeled and paired together as they should be. And then if I just turn my sugar and phosphate into a single like ribbon strand, as having a backbone, this is what we call a DNA ladder, okay? Just to kind of see our bases paired together here. And we normally see this kind of structure when we are asking for those bases matched together, okay? And then if you took this ladder and you just simply twisted it a little bit, that would make your double helix, okay? All right, so I want to remind you guys to return to these slides if you are having trouble recognizing the differences between RNA and DNA, as well as the differences between purines and pyrimidines. Remember today that your nucleotide, that is your building block, that is your basic structure of your DNA molecule. And then finally, don't forget to go back and to read this article if you are interested. This is what it looks like. There are many different women that are on this article that have had some kind of contribution to science, but maybe did not get the acknowledgement that they wanted for that. Okay? Please make sure that you ask me any questions if you're struggling, and go